In this video, we will discuss about the cell fractionation and centrifugation. What is cell fractionation? Cell fractionation, dividing the tissue or cell into its parts or friction, is called a cell fractionation. As you know that cell fractionation, it is the process which is achieved by the process of centrifugation using a centrifuge machine. Cell fractionation is a modern cell techniques in which we can isolate cells and their organelles, for example, nucleus, mitochondria, Golgi complex, etc. etc. The cell fractionation process, as I have told, that it is achieved by the process of centrifugation. So first of all, we will go to explain what is centrifugation. So centrifugation is a mechanical process which involves the use of centrifugal force to separate particle from a solution according to their size, shape, density, and rotor speed. This is known as centrifugation. Then what is a centrifugal force? It is basically a force which travel to move away from the center to the circle is known as centrifugal force. The As I have told that the first step in the centrifugation process or cell fractionation process is grinding of tissues or cell into with the help of a grinder in proper osmotic concentration to form a homogenate. When we form a homogenate, the process is known as a homogenization. So when we grind the tissues of plants or animal or any other living organism in proper osmotic concentration and put them into a test tube, then the test tube is put within the centrifuge machine and spun. As I have told that centrifugation process, first of all, the centrifuge machine start with lighter speed, then into higher speed, and then into much higher speed. So when we spun the centrifuge machine at lighter speed, so two components within the test tubes, they are separated. One is known as the pellet, as you see here in the diagram. So pellet basically, which is also known as precipitate, stand for the concentrated particle in test tube after centrifugation. So the denser and those particles in which their size is large, they separated first and they sedimented at the bottom of the test tube, which can be easily separated. Then it spun again at a higher speed. So then mitochondria, lysosome, peroxisome, rough endoplasmic reticulum, Golgi complex, etc. This type of organelle can be separated at higher speed. When we increase the speed of the centrifuge machine to much higher speed, then the lightest particle, in which their size as well as density are smaller, they will be separated. And their example, as you know that ribosome is one of the lightest particle present in the cells, they will be separated at the last. So these are various steps in which in cell fractionation by the process of centrifugation which is achieved. So we can easily uh, isolate various types of organelles of the cells, larger and denser organelles as well as smaller and lighter organelles according to the speeds of the centrifuge machine. In this diagram or in this picture, you can see the centrifuge machine in which we kept a large number of test tube having grinded tissues in their proper osmotic concentration. If you like the video, please subscribe, share and watch.